Yes. Good afternoon one and all. So, in our previous class we have started with the kingdom Monera, that is we have dealt with bacteria in detail, prokaryotic cell. Okay, in our previous class we have dealt with the bacterial cell structure in detail, its physiology and its metabolic diversity, where we have learnt completely about nutrition and respiration. In nutrition, if we have to look into, bacteria conducts both autotrophic and heterotrophic nutrition. Few of them are photoautotrophic, few of them are chemoautotrophic. Heterotrophic, if we have to take, we have symbiotic, parasitic and saprophytic modes of nutrition. Respiration also it is diverse. Okay, we have aerobic and anaerobic. In aerobic again we have obligate aerobes, facultative anaerobes. In anaerobic we have obligate anaerobes, facultative aerobes. Clear, right? Today we are going to focus on reproduction, the most common modes of reproduction. Okay, most common modes of reproduction, sexual recombination, and then we are going to highlight the useful and harmful bacteria. Okay, so let's see first reproduction during favorable conditions. During favorable conditions, bacteria will mostly undergo the reproduction through binary fission. Binary fission is nothing but just cytoplasmic and nuclear division will happen without any spindle apparatus formation, like mitosis and meiosis, where spindle apparatus will be involved in cell division, but here just there is cytoplasmic division and nuclear division. So, binary fission is without spindle apparatus formation, or there is no role of spindle apparatus here. So, that is why binary fission is also called as amitosis. Okay, binary fission is also called as amitosis. This is most common method of bacterial reproduction, a form of asexual cell division. The bacterial cell divides into two genetically identical daughter cells. Okay, bacteria will divide into two genetically identical daughter cells. Like here if you see the nucleoid, cell wall, cytoplasmic membrane. So, first there will be a small furrow from the periphery towards inside, dividing the nucleus and cytoplasm into two daughter cells. As these two daughter cells coming from a single parental cell, right? So, they are genetically identical to the parent cell. Okay, all the daughter cells are genetically identical to the parent cell. Clear with binary fission? Okay, the major points to be highlighted over here is during favorable conditions the bacteria will undergo binary fission, and there is no spindle apparatus formation over here, and this is also called as amitosis because there is no involvement of spindle apparatus. Next, sexual recombination. Rather than calling it as sexual reproduction, sexual recombination is the most good term to use over here. It is sexual recombination because only single genetic material is involved in the recombination. Okay, so we can best call it as sexual recombination. In bacteria we have three types of sexual recombination. First is conjugation, second is transduction, and third will be transformation. Okay, conjugation, transduction, transformation. First we will see conjugation. Okay, conjugation is nothing but, yesterday we have learnt two different types of DNA that are present in the bacteria. First is what? Genomic DNA or chromosomal DNA. Second type is extra chromosomal DNA, non-genomic DNA or plasmid DNA. So, this plasmid DNA if you take, in this plasmid DNA, F factor will be present. Plasmid DNA will be having what ma? F factor. So, here if you take, see, one bacteria whose plasmid DNA which has F factor is called as F plus strain. Okay, and this is connected to the other bacteria which does not have F strain in the plasmid DNA. So, those bacteria which does not have F strain is called as F minus strain. F plus strain and F minus strain, keep that in your mind. F plus strain is having F factor in the plasmid DNA, F minus strain is not having the F factor in the plasmid DNA. These two are connected through what? Conjugation tube. Okay, these two are connected through conjugation tube. Conjugation tube. This conjugation tube is also called as sex pili. Okay, conjugation tube is also called as sex pili, which connects one bacteria of F plus strain and the other of F minus strain. Okay, so this conjugation tube, which is also called as sex pili, it is also called as true pili. Okay, look at the theory once. Here there is a direct transfer of this F factor from plasmid DNA of F plus strain to the plasmid DNA of F minus strain. So, the direct transfer of genetic material from one bacterium to the other bacterium through the conjugation tube is called as conjugation. This mode of genetic material transformation is called as conjugation. Direct transfer of DNA from donor cell of F plus strain to recipient cell, that is F minus, through cell to cell contact is conjugation. This is first discovered by Liderberg and Tatum in the year 1946 in E. coli, Escherichia coli. What is the mechanism behind this? Okay, first, this requires a sex pilus or a conjugation tube which is connecting F plus strain and F minus strain, and F plus has F plasmid which makes the pilus and transfers the DNA. F minus cell will lack the plasmid. Okay, this pilus will contract drawing the cells together. Pilus will contract drawing the cells together first. DNA will replicate. DNA replicates and passes through the pilus into F minus strain. Okay, DNA replicates and this will pass through what? F minus strain. Donor retains a copy. Donor will be retaining a copy and this is giving to the recipient. So, what mode of replication is happening? Conservation, conservative mode, wherein parent DNA strand will be there, daughter will be there. Both of them will be present, of which parent will be there inside the donor cell only. This will be transferred to the F minus strain. Here there is one more concept to be remembered. Just a second. Yes. So, the transfer of F factor from plasmid DNA of donor cell to the recipient DNA of the recipient cell. So, here if you see, this is from F plus to F minus, whichever we have learnt previously. Okay. If you have to consider one cell which has genomic DNA and plasmid DNA. Okay. In genomic DNA, if we are having this F factor in the genomic DNA, if we have this F factor transfer from the genomic DNA to the plasmid DNA of the F minus strain. Okay. This is called as HFR. HFR strain. HFR strain means what? F factor is present in the genomic DNA. From here, the F factor is being transferred to a recipient cell's plasmid DNA of F minus strain. So, here what daughter cells you will get over here? That is F plus daughter cells. Over here what you get? HFR and F plus. Clear? Understood? So, this is all about conjugation. Any doubt over here? No, right? Yeah. Next is transformation. This is the second mode of sexual recombination, wherein here if you see, which is nothing but the uptake of DNA fragments from the environment by the bacterial cell. Let us assume this. There is a petri dish and a bacteria over which some genetic material is added. So, group of bacteria, they are in competition to receive that genetic material. Okay. Transformation is nothing but the transfer of genetic material or the uptake of genetic material from surroundings or environment by the bacteria is called as transformation. This is given by Frederick Griffith in the year 1928. Okay. Frederick Griffith in the year 1928, who worked out on Streptococcus pneumoniae. He worked on Streptococcus pneumoniae, wherein mechanism involves, wherein DNA is absorbed by the recipient from the surroundings. DNA is absorbed by the recipient cell from the surroundings. This results in genetic alteration which acquires new traits. Okay. So, here can you see the genetic material which is taken from the surroundings, replication will be happening, plasmid DNA of the bacteria that will be combined with the ligase enzyme, the gene from the bacterial cell. Okay. Just remember, conjugation means direct transfer of genetic material from one bacteria to other, from donor cell to recipient. Transformation means uptake of genetic material from the surroundings. So, here comes the concept of competence. One time the question is asked on competence also. Competence is nothing but bacteria which are in competition to receive the genetic material from the environment. Okay? Yes. Next is transduction. Transduction, wherein transfer of genetic material or DNA from one bacterium to another. From one bacterium to another is bacteriophage. Okay? Transfer of DNA from bacteriophage to bacteria. Bacteriophage is what? Virus which is infecting the bacteria, right? So, the transfer of genetic material from this virus to bacteria, bacteriophage to bacteria is called as what? Transduction. Transduction is discovered by Liderberg and Zinder in the year 1951 when they are working on Salmonella typhimurium. Okay. So, here about the bacteriophage infecting bacteria we have learnt clearly in lytic cycle and lysogenic cycle. Okay. So, till here 3 modes of sexual recombinations are clear, right. Once again a quick recap that during favorable conditions bacteria will replicate by what? Binary fissions. In unfavorable conditions sexual recombination will happen. 3 modes we have: conjugation, transformation and transduction. Conjugation is in between 2 bacteria, donor cell and the recipient cell. Donor cell can be F plus strain or HFR strain and the recipient cell is F minus strain. Okay, where conjugation tube or sex pili will be formed between 2 strains and the transfer of F factor will happen. Second is transformation. Transformation is nothing but the uptake of genetic material from the surroundings. Okay, there comes the concept of competence amongst the bacteria to gain that genetic material. Third is what? Transduction. The genetic material which is passed on from the virus to the bacteria, from bacteriophage to bacteria, that is called as transduction. So, here what you have to remember is scientists, their discovery, model organism, okay, and the methods of the sexual recombination and transduction. Conjugation though mechanism is also important. Okay. Now, let us look into the importance of bacteria in humans. Let us work out on the useful and harmful bacteria. First is industrial applications. Okay. First, lactic acid bacteria, which is a popular bacteria, well known bacteria, lactobacillus, which is used in curd preparation. Second is vinegar production, which is done by Acetobacter aceti. Okay. Alcohol production is seen by Zymomonas, Clostridium, Saccharomyces, which are used. Okay. Antibiotics. Antibiotics best examples include Streptomyces gracius, which gives out the antibiotic streptomycin, Bacillus subtilis, bacitracin, penicillium will give out the antibiotic penicillin. Okay. Enzymes which are produced for industrial use, steroid transformations, these are performed by Corynebacterium. Okay. All these are useful bacteria. First the industrial use and next we have learnt on the antibiotics. And then agriculture, nitrogen fixing bacteria. Okay. These are widely used. Just a second. Just a second. So here in agriculture, first is what? Nitrogen fixing bacteria. Nitrogen fixing bacteria, best example we can take cyanobacteria, which is also called as blue-green algae. Okay, cyanobacteria, which is also called as blue-green algae. Nitrogen fixing bacteria, this will convert atmospheric nitrogen into usable forms. First symbiotic, if you take Rhizobium, which lives in the root nodules of leguminous plants. Okay, Rhizobium, which lives in root nodules of leguminous plants, whose main function is what? Nitrogen fixation. Second is free living nitrogen fixing bacteria like Azotobacter and Clostridium. Fine. And next, nitrifying bacteria. Nitrifying bacteria includes what? Nitrosomonas and Nitrobacter. Nitrosomonas will convert ammonia to nitrites. Okay. Nitrobacter will convert nitrites to nitrates. Fine. So, Nitrosomonas ammonia to nitrites, Nitrobacter nitrates to nitrates, nitrites to nitrates. Okay. All the bacteria are important. On what they are working is also important. Next, biopesticides. Bacillus thuringiensis is used to kill insect larvae, and then decomposers. This will help to recycle nutrients in the ecosystem. So, these are the usage of bacteria in the agriculture. And next, medical research. This is used in vaccine preparation. Bacteria are used in vaccine preparation and in genetic engineering as well. E. coli is used as the major model organism for what? Recombinant DNA technology, rDNA technology. E. coli is used as model organism. Plasmids are used as what? Vectors. Plasmids used as vectors. And then, yeah. So, here in different industries, what is the role of useful bacteria and harmful bacteria? Look at the tables keenly. Yeah. Dairy industries, Lactobacillus bulgaricus, which is used in curd preparation and yeast formation. Okay. And then Clostridium botulicum. Clostridium botulicum is majorly involved in food poisoning, which is known as botulism. And then nitrogen fixation, just now we have learnt Rhizobium, which is symbiotic nitrogen fixer, and Azotobacter, those are free living nitrogen fixation, Azotobacter, Azospirillum. Okay. Here harmful, Pseudomonas denitrificans. This is involved in denitrification, loss of soil nutrients. Okay. Next is antibiotic production. Streptomyces griseus is involved in what antibiotic production? Streptomyces. Next, which bacteria are used as biopesticides? This table, if you can remember, it is enough for useful and harmful bacteria. Bacillus thuringiensis, okay, insecticidal protein against the pest. Harmful, Erwinia amylovora, which causes fire blight in apple and pear trees. Next, fermentation. Clostridium acetobutylicum, which produces acetone and butanol. Okay. Harmful include Salmonella typhi, which causes typhoid fever. Okay. And then in genetic engineering, E. coli is used for rDNA technology. E. coli is also having some pathogenic strains which will cause diarrhea and urinary tract infections. Vinegar production. Vinegar production, Acetobacter acetate will convert ethanol to acetic acid. Next, harmful bacteria, Shigella dysenteria, okay, which causes bacterial dysentery. And then bioremediation, Pseudomonas putida, this will break down the oil spills and pollutants. In bioremediation, bacteria is used. Vibrio cholerae will cause cholera disease. Then probiotics. Best example is Lactobacillus only, which is used in curd formation, which is the best probiotic for gut health. Okay. Mycobacterium tuberculosis, which will cause tuberculosis. And then those bacteria which are useful for organic farming, free living nitrogen fixing bacteria like Acetobacter and Azospirillum. Harmful bacteria include Xanthomonas campestris, which causes black rot in crucifers. Okay. Xanthomonas campestris will cause what? black rot and crucifers. Till here it is clear, right ma? Next. Yeah. In genetic engineering, in biocontrol therapy, research tools, cancer research, vaccination development also bacteria are used, bacteria and viruses are used. So, the previous two tables we have dealt with the useful and harmful bacteria. Now, these two tables are involved in useful and harmful viruses and its applications. Look at it carefully. Just a second. Yes. So, here useful and harmful virus and its applications. So, here if you see in genetic engineering, adenovirus, retrovirus is used as vectors. Both adenovirus and retroviruses are used as vectors in gene therapy. HIV, which is human immunodeficiency virus, which will cause AIDS. And then biocontrol and therapy, okay, bacteriophages, which is used to kill drug resistant bacteria, and then TMV, tobacco mosaic virus, which will cause mosaic disease in tobacco, and then research tools, lambda phase, which is the tool in molecular biology and rDNA studies. Influenza virus will cause seasonal flu. Cancer research, in modified viruses which are used in oncolytic therapy for cancer cells, and then hepatitis B, C viruses will cause liver infections, risk of cancer. All these are important ma, just go through the table, read it 5-6 times, you will get it, okay. Vaccination development, cowpox virus is used in smallpox vaccine development, polio viruses will cause poliomyelitis, okay. Then last table for useful and harmful viruses. In plant protection, engineered viruses will carry out beneficial traits in transgenic crops, okay. They will cause beneficial traits in transgenic crops. And then banana bunchy top virus will cause bunchy top disease in banana. And then vaccine production, measles and mumps virus is used in live attenuated vaccines. Rabies virus will cause fatal neurotrophic virus. And then gene delivery, okay, recombinant adeno associated viruses, which is efficient gene delivery tool, okay. Harmful viruses include dengue virus, which cause dengue fever, coronavirus SARS-CoV-2 variant, which caused COVID-19, and rhabdovirus causes rabies in animals and humans, okay. With this, from microbial world, we have dealt with bacteria and viruses totally. With today's class, bacteria and virus class is done. In our next class, we are going to start with Thallophyta, and then we are going to look into fungi, and then Bryopterido, okay. So, with this we can end this concept over here. Yes, thank you.